GRIAS Academy.
100 days, 100 teams, one goal, UPSC Prelims. Hello all, welcome back to the program Mission Surya Phase 3 STEM Revision Program, where we deal with science and technology and environmental topics completely regarding the Prelims perspective. Okay, let's get started today's session. In today's session, we are going to see few terms which are related to biotechnology. They are coming in use frequently. Let's see what are those terms now. See the few terms like next car 19, RNAi, BRR Dhan 100, TNB, TNPB, OOC. These are few biotechnological terms we are seeing. Next car 19 is about cancer vaccine which is developed indigenously in India, right? Next, RNAi is about gene silencing technique, or we can call it as RNA interference. Next, BRR Dhan 100 is about rice varieties which are produced using biotechnological methods. TNB is regarding few protein synthesis. Okay. Next, OOC is organ on chips. These are few biotechnological topics which are being seen in the news frequently. So let us see each one of the topic detailedly. Okay, let's move on to the topics now. Moving on to the first topic, that is next car 19. So what is this next car 19? It has made a significant leap in indigenous cancer treatment with next car 19, providing an affordable and highly effective solutions for specific blood cancers. Specific blood cancers like leukemia and lymphoma. These are few these two are blood related cancers. Okay, now let us see the key features of next car 19. What it is? It is developed in India indigenously. It is not imported from somewhere or so. It is completely made in India. That's why we are calling it as indigenous. Next, indigenous CAR in the sense chimeric antigen receptor T cell therapy. Okay, which diseases it is targeting? It is targeting specially developed for B cell blood cancers. What are these B cells? B cells are typically the white blood cells. Right? Typically these are the white blooded cells which are responsible, type of white blood cells responsible for producing antibodies in the immune system. Producing antibodies in the immune system. Okay. The cancer which is caused by B cells is mainly targeted. Okay. Now, let us see how this CAR T cell therapy is used in the cancer treatment. First of all, what happens is the blood is collected from the patient. And from that blood the T cells are separated. You can see it here. The T cells are separated and provider returns remaining blood. The remaining blood which is left over, which is given back to the patient again. Then these T cells are genetically altered specific special receptors called chimeric antigen receptors are binded to these T cells, which now lead to millions of CAR T cells are grown in the in vitro process, that is in the lab. The conditions which we grow in the lab are called anything, not only this T cells. Any cell which is grown in the lab, those conditions are called in vitro conditions. Okay, so millions of CAR T cells are grown with these receptors binding to the T cells, and then before giving the therapy, before giving the CAR T-cell therapy to the patient undergoes the general chemotherapy process of cancer. Then the patient will be given the T-cells introduced into the bloodstream, which fights with the cancer-causing agents in the body. Okay, this is what about the next CAR-19, the T-cell therapy, CAR T-cell therapy. Now, let us look what are the developers. Who developed it? Means it is developed by ImmunoAct. ImmunoAct, it is a company incubated in IIT Bombay in collaboration with Tata Memorial Hospital. What is the government support? From government, which department is supporting is? It is related to biotechnology, so the Department of Biotechnology is backed up by Department of Biotechnology and the Biotechnology Industrial Research Associate Council. BIRAC plus Department of Biotechnology are supporting this one. Okay, next. Regulatory approval. Who is giving this approval for this vaccine to come into the market? It is granted market authorization by the Central Drug Standard Authorization in 2023. Okay, next. See, there are few mechanisms you have to know. I have already told you about the CAR T-cell therapy. See, it is a type of immunotherapy. That modifies the patient's own immune cells. T-cells are the immune cells. Specifically, it alters T-cells. A type of white blood cells primarily responsible for cytotoxic functions. Meaning, they kill infected or abnormal cells. Okay. Now, let us look into the process. As I told you, the T cells are extracted from the patient, genetically engineered in lab, that is the in vitro conditions, to produce receptors called chimeric antigen receptors. These receptors are produced and then reinfused into the patient back again through the bloodstream. Okay. These newly minted CAR T cells can now specifically identify, bind to and destroy the cancer cells in the human body. Okay, this is what the process. You can see it here. See, the T cell, the chimeric antigen receptor which is bind to the T cell, which it becomes like this, right? And it fights against the cancer cells which are responsible to create the blood cancer in the human body. Okay, next. Let's see a few advantages of this therapy. Sustained action in the sense, unlike the traditional medicine like chemotherapy, generally cancer patient undergoes chemotherapy, which is a very painful process that metabolizes and leave the system. Just what happens in the chemotherapy, it just metabolizes the system and leave. There is no cure for cancer in previous days now, but we are coming up with vaccines for cancer. Okay, CAR T cells, they act as a living drug in the body, means they possess the body for long-term protection against the cancer relapse. Okay, next. Faster recovery. Compared to the chemotherapy therapies and all, this gives us the faster recovery, means a shorter treatment duration compared to aggressive chemotherapy or stem cell transplantations. Next, affordability. Coming to the affordability, significantly lower cost barrier compared to the previous which are highly expensive because they are mostly imported. We mostly imported this cancer vaccines and all, but now as it is developed indigenously in India, the cost also has come down, making it accessible to a larger demographic in India. Okay, now these are few advantages, and let's see few challenges and side effects of this therapy. High specificity means therapy designed for only specific type of cancer, will not work for any other cancer. Means generally this next CAR-19 is developed only to treat blood cancer, not any other cancers in the body, specifically targeting the B cell type cancer. Okay, so this is one drawback. We can see cytokine release syndrome basically after this, means a severe systemic inflammatory response, caused by overactivation of immune cells. What are we doing in this therapy? We are activating the immune cells, right? So overactivation of these immune cells may lead to cytokinin release syndrome. Okay, this word is very much it is frequently tested keyword for UPSC. So make a note of it. Okay. Next, neurotoxicity. Neurotoxicity means which can have a temporary or severe negative effects on nervous system. Any effects on nervous system, it is neurotoxicity. Next, immunosuppression. Immunosuppression means it carries a higher risk of secondary infections. Many secondary infections may be caused. These are few drawbacks of this therapy. Okay. Yeah. Moving on. Let's see one practice question about this concept. See the question. Consider the following statements regarding next CAR-19 recently seen in the news. The first statement is given like, it is India's first indigenously developed CAR T-cell therapy. Second statement, it is designed as a universal treatment for all types of solid and liquid tumors. Third statement is, it was developed with the support of Biotechnology Industry Research Assistance Council. Okay. Which of the statements given is above is or are correct? Option A, 1 and 2 only. Option B, 2 and 3 only. Option C, 1 and 3 only. Option D, 1, 2 and 3. So what would be the correct answer? The correct answer would be option C. Why? Because yes, it is in first indigenously developed CAR T-cell therapy, that is correct. See the second statement, it is designed as a universal treatment for all types of solid and liquid tumors. Is it designed for all types of cancers in the body? No, it is specifically designed only for few blood cancers. So the statement number 2 is wrong. The statement 2 is wrong. Coming to statement 3, it was developed with the support of the Biotechnology Industry Research Assistance Council. Yes, it is supported by Biotechnology Industries Research Assistance Council plus Department of Biotechnology. Okay, so only option statement 1 and 2 is 1 and 3 is correct. Statement 2 is wrong. So the option is C, 1 and 3 only. Okay, now done. Let's move on to the next topic. Our next topic is RNA interference or gene silencing, right? RNA interference and gene silencing. Basically in UPSC what happens is the examiner might trick you with few concepts with like gene silencing, gene targeting, gene sequencing, gene silencing, gene targeting. All these few conceptual words the examiner might trick you. So have a look on those. Okay. Now see what is RNAi. RNAi is, it is a crucial biotechnological tool where like as I said you, UPSC frequently may ask the conceptual clarity between gene editing, gene silencing and all. So have a look on it. Okay. Let's see the core mechanism. It works on at the RNA level, not at the DNA level. Instead of altering the organism's underlying DNA, it intercepts and destroys specific messenger RNA before it can be translated into proteins. Basically in the translation process, in the transcription process, what happens? The double stranded DNA means the gene becomes RNA by an enzyme RNA polymerase. Then this RNA is converted into a mRNA, that is messenger RNA. This messenger RNA is converted into proteins. This is what the basic pattern is. Okay, done. So what happened here? What happens in this mechanism is it intercepts and destroys specific messenger RNA. It specifically destroys the specific messenger RNA before it can be translated into proteins. Before translating into proteins, these mRNAs are targeted by RNAi. Okay. Now, what is the natural function? It acts as a internal cellular defense mechanism. Internal cellular defense mechanism which acts against the viral infections and regulates normal gene expression. Okay. Now, moving forward. There are few enzymes which you have to have a look on it, because like UPSC might frame questions based on these biological components. So you can have a note of it. Like, see the first enzyme, that is the trigger. The trigger is dsRNA, that is double-stranded RNA. We generally learn about double-stranded DNA. Now it is double-stranded RNA. Double-stranded RNA means this process begins when the cell detects the double-stranded RNA, which is often a sign of viral invader. Which is a sign of a viral invader in the body. Next, dicer enzyme. Dicer enzyme is like a scissor which cuts the gene. Like this enzyme cuts the double-stranded RNA into tiny fragments, into small tiny fragments known as small interfering RNA. Small interfering RNA or micro interfering RNA. Micro RNA. Okay. This is about dicer enzyme. Next, we see RISC complex. RISC complex, it is a group of enzymes, not a single enzyme, it is a group of enzymes. This is the tracker. This is the tracker. If a gene is there, see, this is a gene which are coded with all the amino acids. A, T, G, C. A, T, G, C. These are the amino acids where the gene is coded with. Okay. Now, the trigger point is the double-stranded RNA. Now, this is double-stranded RNA. Okay. Now, this enzyme, it acts as a scissor. If I wanted to cut a gene, I'll use another pen. If I wanted to cut a gene here, it acts as a scissor. Now, this double-stranded RNA is cut into small slices, small pieces by using this dicer enzyme, which are small interference RNA, small interfering RNA or micro RNA. Now, this group of enzymes which are called RISC complex, this is the tracker. This is like a tracker which tracks which position have to be tracked. In the sense, the small interfering RNAs bind to a group of enzymes. All the small pieces, and the pieces which are cut by the dicer enzyme will come and bind to the group of enzymes called the RNA-induced silencing complex. RASC means RNA-induced silencing complex. Then the small interfering RNA acts as a guide to help this RISC complex to find the matching target mRNA in the cell. Okay. Now, agronaut protein. What is this protein? This is an executioner, like a crucial protein within the RISC complex. A protein will also be in this. It is a crucial protein, which is agronaut protein, that physically cleaves the targeted mRNA, effectively silencing the gene. Effectively, which silences the gene. Next. This is some conceptual clarity of the enzymes. Next. Moving on. Okay. Advanced gene silencing variants. We'll see what are those. DNA based RNAi, which is an artificial short where DNA sequence designed to express double-stranded RNAs, which then activate the cell's natural RNAi pathway. Next. See, the second one is CRISPR-mediated RNAi. CRISPR-mediated RNAi is DCAS9. DCAS9. It is CRISPR interference. CRISPR is nothing but a gene editing tool, like the dicer enzyme which cleaves the gene. Uses and modified dead Cas9 enzyme. It uses dead Cas enzyme. It binds to the target site to block transcription. Where the transcription process has to be blocked, where mRNA is converted into protein. here it has to block, the transcription process has to be blocked to get the described whatever we want to get the whatever we are wanting. Like if you wanted to develop a what you call basically the previous thing. Take an example of the cancer therapy vaccine, where it has been developed indigenously by taking the T cells, and it is bind to the CAR, right, chimeric receptor and antigenic receptor, right. So this is developed in vitro, basically using this gene silencing and non-transcriptional process, and anything to produce the transcription process is common for everything. Okay. Now initially in the mRNA levels only this transcription has to be blocked. This is done by dCas9, right? But it does not cut the DNA. See here, it uses a modified dead Cas9 enzyme. It binds to the target site, to block the transcription, but does not cut the DNA. Next, Cas13 enzymes. What these enzymes do is, unlike traditional Cas9, means they usually target DNA, right, which targets DNA. Cas13 is a CRISPR associated protein. CRISPR associated protein that specifically targets and leaves RNA. Basically this Cas9 which targets the DNA, but Cas13 which targets and leaves the RNA specifically. Okay. Next, we can see a few prelims trap in this. Like traditional CRISPR-Cas9 operates at the DNA level, which is gene editing, and does not fall under RNA interference. So please look into it. The examiner might trick you with Cas9 and Cas13. Okay. Next. See few applications. This can be used in this RNA interference can be used in various sectors like agriculture, in healthcare, in cancer therapies, in few genomic functions, and few therapeutics, and all. Major applications are there for this RNA interference with gene silencing. Let's see in agriculture. In developing pest-resistant crops and engineered traits. Example, decaffeinated coffee plants can be produced by using this technology, because it is only thing is, when a gene is produced, generally a coffee plant to grow, it has caffeine in it, right? To decaffeinate that plant, we can use this gene silencing technique, and we can develop without caffeine content in that. Okay. So that we are targeting the caffeine content, we are deactivating the caffeine. So we can do it by this gene silencing technique, that is RNA interference. In therapeutics, how developing treatments for viral infections like HIV, hepatitis B, and all, hepatitis C, and respiratory diseases by silencing the viral gene. We can silence the viral gene. Okay. Next, in cancer therapy, how? By suppressing the mutated genes responsible for cancer cell growth. Next, few functional genomics, like quickly determining the functions of newly discovered genes by turning them off and observing the results. We can use it in the genomic process also. These are few advantages. Now, let us see one practice question for this. It is, with reference to RNA interference, consider the following statements. Statement 1. It is a natural process that edits the DNA of cell to prevent the replication of viruses. In second statement is, the enzyme dicer plays a crucial role in cleaving double-stranded RNA into interfering RNAs. Next, third statement is, it has applications in developing pest-resistant crop plants. So, which of the following statements given above are correct? Means option A, 1 and 2 only, option B, 2 and 3 only, option C, 1 and 3 only, option D, 1, 2 and 3. So, what is the correct answer? The correct answer is 2 and 3 only, means option B is correct, right? Okay. So what is it? Option 2 and option B is correct, that is statement 2 and 3 are correct. Why? Because it is an see the first statement. It is a natural process that edits the DNA of a cell to prevent the replication of viruses. Friends, make a have an idea on it. RNA interference works only in the RNA level, not in the DNA level. It alters in the DNA level, not it alters only in the RNA level, not in the DNA. Okay? So it is not DNA, it has to come here RNA. Okay? Next, the enzyme dicer plays a crucial role in cleaving double-stranded RNA into small interfering RNAs. Is it true? Yes, it is, because the dicer is it works like a scissor to cut the double-stranded RNA. Next, it has applications in developing pest-resistant crop plants. I already told you in the advantages, it is used in the agriculture for developing pest-resistant crop plants. Okay? Now, let's move on to the next topic. Our next topic is DRR than 100. It is about the rice varieties. Okay? It is a breakthrough from ICAR, Indian Council of Agriculture and Research. See, it is developed using SDN1 genome editing in popular Samba Masuri rice. This high-yielding variety is a game changer for rice cultivation. What are the key features of this? Means 19% increase in grain yield, 19% increase in grain yield, 15 to 20 days earlier maturity than comparatively to the original Samba Masuri rice, and moderate drought tolerance. Not much. It doesn't affect for much drought. It's a moderate drought tolerance. Retained grain quality. It has a retained grain quality. These are few key features for developing this DRR Dhan 100. Okay, which is developed by Dr. Satyendra K. Manguthai, Dr. M. Sundaram and team, which is in ICAR, Indian Institute of Rice Research, Hyderabad. Okay, it is in Hyderabad. So, this is the world's first genome edited, world's first, not India's first, it is world's first genome edited rice. It is Kamla and Pusa DST rice 1. Okay, these two names are given. The Indian Council of Agriculture and Research, ICAR, has successfully developed the world's first genome edited rice varieties, making a monumental shift in climate resilient agriculture and biotechnology in India. Okay, now what are the breakthrough varieties now? India has introduced 2 distinct varieties. What are those? Developed without using any foreign DNA, which are developed using without any foreign DNA. First one is the Kamal, right? Kamal, how it is developed? It is developed by ICAR, Indian Institute of Rice Research, Hyderabad. Okay. What is the parent variety of this Kamala? It is Samba Masuri. Samba Masuri, basically everyone would be knowing this is the rice variety which commonly used. So, using this, the Kamala is being used. Okay, this is the parent variety. Now, which gene is targeted? Cytokinin oxidase 2. This gene in Samba Masuri rice, this cytokinin oxidase 2 is targeted to edit and produce Kamla variety. Now, what are the key traits? Earlier maturity means it saves water by requiring 3 fewer irrigations. Superior yield, in the sense 19% increase in the yield. Drought tolerance, moderate drought tolerance, and high nitrogen use efficiency. It can use high nitrogen rather. Okay. These are few points about Kamala variety of rice. Next, let's see the next variety. The next variety of rice is Pusa DST rice 1. Right? It is developed by same Indian Agricultural Research Institute, New Delhi. That Kamala variety is developed in Hyderabad. Right? See, don't get confused about it. Kamla variety is produced in Indian Institute of Rice Research, Hyderabad. Indian Institute of Rice Research, Hyderabad. Whereas Pusa DST Rice 1 variety is developed in Indian Agricultural Research Institute, New Delhi. Okay, next. What is the parent variety for it? Parent variety is MTU 1010. This is the variety, rice variety. Which the gene is targeted? Drought and salt tolerance gene. Drought and salt tolerance gene. Next, what are the key traits? It is specifically engineered for enhanced yield under extreme stress conditions, means without water. It can grow in minimal conditions without less water, because rice needs more water crop, right? So inland salinity, coastal salinity and alkaline soils, because as Mumbai is in the coastal area, these genes are targeted, basically drought and salt tolerance genes. Okay, so that this variety can grow in few extreme stress conditions. Okay, got it? Next, see the core science of it now. Genome editing, this is genocrops, genetically modified crops. Genetically modified crops in the sense which are developed, these crops which are mutated or developed for bearing in certain conditions. Okay, which have grown in vitro only, with this gene splicing, silencing, this gene editing or targeting a specific gene, everything is done in vitro only, then only they are planted and all. Okay, basically another one, UPSC frequently tests the conceptual boundary between genome editing and genetically modified crops. Okay, let's see what is genome editing. Let's see what is genome editing. Genome editing means, I have already told you, it is like a molecular scissor which cuts the gene, like CRISPR-Cas9 to make precise targeted changes, means deletions, any deletion of gene or any insertion, this has to be there. I need a drought example, I need a drought tolerance plant. So I induce a gene, I insert a gene which is responsible for a crop to tolerate the drought. And like that. So, to make precisely targeted changes like deletions, insertions to the plant's existing DNA, where no foreign genetic material is introduced. Okay, next, genetically modified. What is genetically modified? Genetically modified crops means it involves inserting a completely foreign gene. Okay, inserting completely foreign gene from another organism. Example, introducing a soil bacterium gene into cotton to make BT cotton. Everyone might be knowing BT cotton, BT brinjal, everything. These are genetically modified, means completely a foreign gene from another organism is taken and introduced into the of another organism. Sorry. It involves inserting a completely foreign gene. A completely foreign gene from another organism is inserted into the DNA to completely modify it. Like see, introducing a soil bacterium into the cotton to produce BT cotton. Okay, this is one example. Next, the technology used. What is the technology used? SDN1. Both new rice varieties were developed using site-directed nuclease 1 technology. Site-directed nuclease 1 technology. It uses nuclease, enzyme nuclease, to create a targeted break in the host DNA. To create a targeted break in the host DNA, which the plant repairs naturally. It does not introduce any foreign genetic material and is biologically indistinguishable from natural mutations. Natural mutations means if a flower is growing, if a plant is growing, if a flower comes out or a leaf or a flower. See, in hibiscus we can see it basically. If a red hibiscus is growing, basically due to few climatic conditions and environmental factors, what happens is the red color of hibiscus turns into some mutate some another color, like it could be red to a half, pink to the other half. This is called mutation, a sudden change in the organism or sudden change in the plant. This is called mutation due to few external factors or the internal factors. Okay, this leads to the mutation. This is called mutation. Okay. What is the regulatory framework? Who is regulating all this? This is regulated by, because genome edited crops using SDN1 technology and SDN2 technology do not contain foreign DNA. It do not contain foreign DNA. India has relaxed its regulatory framework for them. Okay. Now, what is the exemption from this? The exemption is under the Environment Protection Act 1986. Under Environment Protection Act 1986, crops developed using SDN1 and SDN2 technologies are exempted from the strict GMO regulations, which is rules 7 to 11, that govern standard transgenic genetically modified crops. Okay. Who are the approving bodies for genetic engineering crops? Yes, these lines are cleared by Institutional Biosafety Committees, IBC, and the Review Committee on Genetic Manipulation. Review Committee on Genetic Manipulation, bypassing the lengthy GAC, that is Genetic Engineering Appraisal Committee. The trials required for genetically modified crops. All these institutions will act as the approving bodies for the genetically engineered crops. Okay, next. Let's see a practice question for this. With reference to the regulatory framework for biotechnology in India, consider the following statements. Statement 1. Crops developed using site-directed nuclease 1 SDN1 technology are subjected to the exact same stringent field trial regulations as traditionally genetically modified crops. Statement 2. Genome edited crops utilizing SDN1 technology do not contain any foreign DNA. Statement number 3. The Review Committee on Genetic Manipulation is involved in clearing genome edited crop lines in India. Which of the following statements is or above are correct? Option A 1 and 2 only, option B 2 and 3 only, option C 1 and 3 only. So, what is the right answer? So, what is the answer for this question? The answer is 2 and 3 only. Option B is correct, because see, crops developed using site-directed nuclease 1 technology are subjected to the exact same stringent field, see this word, exact same stringent field trial regulations as traditional genetically modified crops. No, statement 1 is wrong, because SDN1 technology and SDN2 technologies which are there, which are not exact same stringent field trials regulations. Okay, these are not straight. Now, few regulations, for these genetically modified crops, few rules have come up now. So, they are not targeted to the same stringent field trial regulations. Field trial regulations have been levered by the ICAR. Next. Genome edited crops utilizing SDN1 do not contain any foreign DNA. Yes, it doesn't contain any foreign DNA. Statement 3. The Review Committee on Genetic Manipulation is involved in clearing genome edited crop lines in India. Yes, this statement is also correct. So, only the first statement is wrong, other two statements are correct. So, the option is B, 2 and 3 only. Now, let us move on to the next topic. Our next topic is TNPB, means Indigenous TNPB gene editing technology. Indigenously brought up gene editing technology. Okay, what is it? The Indian Council of Agriculture Research ICAR has developed a patented and indigenous miniature genome editing tool. Miniature genome editing tool based on TNPB proteins. Okay, using this TNPB proteins, the ICAR has developed the indigenous miniature genome editing tool. Okay, Next, this serves as India's answer to the globally patented expensive CRISPR-Cas system. CRISPR-Cas systems are very expensive. Rather than this, we have indigenously developed a genome editing tool called TNPB gene editing technology using TNPB proteins. Okay. Now, what is TNPB technology? The mechanism is just like CRISPR-Cas9. CRISPR-Cas9, what we have learnt it before, CRISPR-Cas9 used as a scissor. It edits the gene, it cuts the gene, okay, DNA or RNA, whatever it might be, okay. TNP acts as a molecular scissors. It is guided to a specific DNA sequence. Guided to only specific DNA sequence. If I want the sequence to cut from amino acid A to amino acid G, so you have few regions: promoter region, gene region, 16S ribosomal region and all. If I want to cut the gene at a ribosome level, I'll give the orders to cut the gene at the ribosomal level. Now this TNP acts as a molecular scissor. It goes near to the ribosomal zone and it cuts the gene there. Makes a precise cut, allowing the cell's natural repair machinery to insert, delete or modify the genes. This is how TNP gene editing tool is worked. Okay. Now, let us see what is the source organism for it. The tool utilizes TNPB derived Deinococcus radiodurans. Deinococcus radiodurans is an organism basically. This is the source organism, a bacterium which is highly resistant to radiation. Highly resistant to radiation like UV radiations. It can highly tolerate the few environmental conditions. It can highly tolerate basically complete extreme dryness. Extreme dryness also it can handle. It can also resistant to UV radiation. Obviously it is also resistant to the oxidative stress also. Okay, this Deinococcus radiodurans is a bacterium which is highly resistant to radiation. Like being humans, we can expose to radiation for a few extent, not like that. Millions and 100 times than us, this bacterium can sustain the radiation. Okay, next. Deinococcus radiodurans is an extremophile bacterium known for its extraordinary ability to survive extreme radiation, cold, dehydration and acid. Okay, it is harmless to humans. And often used in bioremediation, cleaning up radioactive waste. To clean the radioactive waste also, this is being used. Okay, this organism is used. Now let's see the difference between TNP versus CRISPR-Cas9. These two are also molecular scissors only, where the core difference is it is most likely area for statement based questions. So they might ask TNBP, difference between TNBP and Cas9, CRISPR-Cas9. Okay, see the size. Coming to the size, the miniature is the advantage. This TNPB gene editing tool is very small, a miniature tool. Okay, where significantly smaller, like 400 to 500 amino acids only they have. But in CRISPR-Cas, what happened? Cas9, it has 1000 to 1400 amino acids. Compare how big it is and compare how TNPB is smaller than this CRISPR-Cas9. Okay. Next, delivery mechanism. Because of its compact size, TNPB can be easily delivered into plant cells via viral vector, using viral vectors. This bypasses the need for complex, time-consuming tissue culture-based delivery systems required for larger CRISPR molecules. Okay, basically to introduce this CRISPR molecules into the plant, we generally need plant tissue culture techniques to be happened in vitro. Then only we can do it. But compared to TNB, when coming to TNPB, it is smaller in size, right? Because of that miniature size, it can use a viral vector to induce into the plants. No need of any time consuming tissue culture techniques and all. Okay, next. See, the intellectual properties, IPRs. CRISPR technologies are heavily patent globally, not indigenously. They are patent globally. Example, by the Board Institute and the Koratava Agri Sciences, meaning commercial cultivation of CRISPR edited crops in India requires steep licensing fee. By using this CRISPR-Cas9 and all, the India requires steep licensing fees, whereas TNPB is indigenously patented by ICAR. It is indigenously patented by ICAR. No, it is not patented globally. Okay, offering absolute IP freedom and drastically lowering the cost of developing genome edited crops. So genome edited crops basically they cost heavily because we have to pay a fee, steep licensing fee. Okay, now it is not required. We have TNPB genome editing tool, so that we are free to pay this. We are free from paying steep licensing fee in the intellectual property rights. Okay. Next, see what is GM and GE, genetically modified versus genetically engineered. Genetic modification is, it introduces foreign DNA from a completely different species or different organisms. Example, BT cotton, BT brinjal and all. It creates a genetically modified organisms. What is gene editing? Gene editing is, it makes precise changes, means the insertions or deletions to the organisms existing DNA using tools like CRISPR or TNB. No foreign DNA is added in this. But for genetically modified crops, foreign DNA is used, completely foreign DNA is used from different species. Okay, have this thing. Next, let's see one question. Practice question is, consider the following statements regarding the newly developed TNPB gene editing technology in India. Statement 1, it is a miniature genome editing tool that acts as a molecular scissor similar to CRISPR-Cas9 system. Statement 1 is that, statement 2 is, unlike the CRISPR-Cas system, the compact size of TNB prevents it from being delivered via viral vectors relying exclusively on tissue culture methods. Now, statement 3, it has been derived from an extremophile bacterium known for its high resistance to radiation. So, which of the following statements given above are correct? Option A, 1 and 2 only. Option B, 2 and 3 only. Option C, 1 and 3 only. Option D, 1, 2 and 3. What is the correct answer now? The correct answer is option C. Right? Am I right? Option C is correct because it is a miniature, first statement, it is a miniature genome editing tool that acts as a molecular scissor similar to the CRISPR-Cas9 system. Yes, it is true. It acts as a molecular scissor unlike CRISPR-Cas9 system. Next, unlike the CRISPR-Cas system, second statement, unlike the CRISPR-Cas system, the compact size of TNPB prevents it from being delivered via viral vectors relying exclusively on tissue culture methods. No, the statement is not completely opposite. The CRISPR-Cas9 technologies generally needs a tissue culture monitoring method. Rather, TNPB gene editing tool requires a viral vector due to its miniature structure. Okay, so it is given in an opposite way. So, statement 2 is wrong. Statement 1 is right. Next, seeing the statement 3. It has been derived from an extremophile bacteria known for its high resistance to radiation. Yes, this is true. Okay, this is the source organism, right? We have seen. Yeah. So, option C, 1 and 3 only is correct. Okay. Now, let's move on to the next topic. Our next topic is organ on chip. Now, moving on to our next topic. Our next topic is organ on chip. Organ on chip means, see, the organ on chip market, The organ on chip market is rapidly expanding, aligning closely with India's newly introduced Bio E3. India has introduced Bio E3. What is this Bio E3? Biotechnology for economy, environment and employment. Bio E3 is biotechnology for economy, environment and employment. It is a policy to revolutionize precise medicine and reduce dependency on animal testing. To reduce the dependency of drugs or anything on the animals, this organ on chip concept has been come up. Okay. Now, see. Let's see what is an organ on a chip. OOC is a micro-scale 3D culture system. A 3D culture system that is designed to stimulate the physiological and medicinal, mechanical environment of human organs. Means on a chip they are creating liver. Chip in the sense microchips, not a big one. Microchips, liver, heart, like what you say, lungs. These are created on a chip. Okay, that's why we call it as organ on a chip. So, what is the core technology? Basically, this works on the technology. Basically, it works on microfluids. Generally, for a heart to pump and all, it requires blood, right? So, it needs the flow of blood and all. So, those are also being given to the organs on chip through microfluids technology. It relies heavily on microfluids, the use of tiny channels to manage the continuous flow of fluids, nutrients and oxygen, mimicking human blood flow around the arranged living cells. Okay, same how our heart functions in the human body, at the same way it is mimicked on the 3D chip, that is organ on chip. Okay, whatever it requires, the nutrients, the oxygen, the blood flow, the blood, everything, water, oxygen, nutrients, whatever is required for an organ to function, everything is given through microfluids, so that the channels are created so that the organ works normally as it works in the human body. Okay, same, the mimic of our human body, they are preparing the organ on a chip. Okay, what is the goal? Why it is created, why these organs on chips and all, why all these things are coming up? Means the main goal is to replace traditional 2D cell culture and animal testing. With highly accurate human tissue models for drug testing and disease modeling. Generally, for any drug testing and disease modeling, we used to practice animal testing, right? Basically on animals like rats they used to do, and then they are given to the humans once they are good enough. Okay, to reduce all those things, organ on chip concept has come up. Okay, next. Understanding the new approach methods. Let's understand few new approaches of methods. That is, organ on chip falls under a broader category of modern biomedical technologies known as new approach methods. Let's see few. Organoids. What are these organoids? Organoids are 3D structures which are created by growing cells often from the embryonic or induced pluripotent stem cells in a defined environment to mimic the tissues. Okay. Next, spheroids. What are spheroids? These are 3D cell clusters that are particularly highly effective at mimicking tumor behavior compared to flat 2D cultures. Next, bioprinting. Bioprinting is using 3D printing, means additive manufacturing to physically build living tissues layer by layer. Okay, these are few new approach methods. Next, have a note on it. Few UPSC may ask questions relaying to difference between these emerging biotech terms and all. Okay? No. Why is organ on chip a game changer? Because in precision therapeutics, it allows for testing how specific drugs affect unique tissue environments. Advancing precision medicine means tailoring treatments based on genetic, environmental and lifestyle variability rather than a one-size-fits-all approach. Okay, next, ethical alternative, because as drastically it reduces the need for animal testing in the preclinical drug trials, preclinical stages of drug development. Next, complex modeling. It is capable of linking multiple chips together. It is capable of linking multiple chips together, like a lung, a heart, kidney, liver. This all. It can link the multiple chips together to observe multi-organ interactions and systemic side effects. By using a drug, any malfunctionings are happening or what, any which organs are being disturbed or any side effects are being come. All these are seen by this organ on chip technology by linking these chips together, like liver on a chip connected with heart on a chip. These two can be connected and can see if these two organisms when they are working on the same this thing, when these two are working, any effect is happening in between these two, or any damage is causing or any side effect is happening, all this can be monitored. Okay, next. Basically all this used to be happen on animals. Now to control that, we have brought up organ on chip. Now, who is the regulating bodies and policy steps? Amendment to the new drug and clinical trial rules 2019. India legally permitted the use of human organs on chips and other NAMs as an alternative or supplement to animal testing for evaluating new drugs. By which one? Amendment to new drug and clinical trial rules 2019. Next, BioE3 policy. BioE3 policy is a national-wide framework to drive biotech innovation with a core focus on precision therapeutics. Next, Phenomenon Indian project, which is led by CSIR, Council of Scientific and Industrial Research. This aims to generate a comprehensive phenomena database tailored specifically to the Indian population and advanced precision medicine. Okay, next, Genome India project, GIP, a government-led initiative. It is a government-led initiative to map the genetic diversity of India to help anticipate and modulate diseases. Next, coming to the global context, the USA passed the FDA and FDA Modernization Act 2.0 2022, officially allowing researchers to use organ on chip as a valid alternative to animal testing for preclinical drug trials. Okay, now USA also passed a FDA for using organ on chip. Okay, next here normal practice question for prelims. See, question. Consider the following statements regarding organ on chip technology. First statement is, it is a biological technique that completely replaces damaged human organs by implanting microchips directly into the patient body. Second statement is, it utilizes microfluids to stimulate human blood flow and mechanical conditions on a miniature scale. Your third statement is, in India, recent amendments to the new drugs and clinical trials rules permit the use of OOC as a preclinical alternative to animal testing. Which of the following given above statements is or are correct? Option A is 1 and 2, option B is 2 and 3, option C is 1 and 3 only, option D is 1. So, what is the correct answer? The correct answer is option B, that is 2 and 3. Because see the first statement, it is a biological technique that completely replaces damaged human organs by implanting microchips directly into the patient's body. No, this is not true because we are not using it for organ transplantation. Not using for organ transplantation, rather these are organs on chips are used for In vitro process. For the research pre-clinical trials rather than animals. So, statement 1 is wrong. Statement 2 is correct because it utilizes microfluids to stimulate human blood flow and mechanical conditions on a miniature scale. Next, option 3, in India, recent amendments to the new drug and clinical trial rules permit the use of OOC, like organ on chip, as a preclinical alternative to animal testing. Yes, this is also true. So, statement 2 and statement 3 are correct and statement 1 is wrong. Okay. So, that's all for today. These are the topics. To have a complete note on it, please do visit our website and download the notes from the website. Do follow and like our YouTube channel. Do subscribe. Do follow our Telegram channel and the Instagram channel as well for your UPSC preparation. All the links are provided in the description box. So do follow our session and let's see you all within tomorrow another new topics. Thank you.